Good morning, everyone. In this session, we will discuss about preparation of different reagents that are specific for fish analysis. Before we start reagent preparation, we will give a brief overview of different equipments and consumables required for fish. This is a table showing different equipments that are specific for fish. If we can recall that fish stands for fluorescent in situ hybridization, and here we measure fluorescent signals in different biological samples. So obviously we need a fluorescent microscope. Since we are working in situ, that is over a glass slide, and the main principle of fish is based on hybridization. So we do need a hybridization chamber. Other equipments that are specific for fish analysis include water bath, hot plate, which is preset at 40 degrees Celsius. We have a different session on slide preparation where we will talk in detail about this instrument. pH meter, which is an essential equipment in the fish processing as we are working with different buffers having specific pH. And lastly, magnetic stirrer with hot plate. It is desirable to have this equipment. However, it is not mandatory to have this equipment in a fish lab. Apart from these equipments, few other equipments as shown in this table is also required. However, the list is extensive and we have included few of these equipments. These equipments are also used in some other labs. Now we will show you some pictures of few of these equipments. This is the image of a water bath. Next image is that of a magnetic stirrer with hot plate. In the inlet, we can see a solid white capsule-like structure, which is the magnetic stirrer, which is placed on a beaker, which is then placed over this hot plate, which will help in easy solubility of the salt in the solution. And as I have said, it is better to have this instrument, but it is not an absolute essential. This is a pH meter. The vertical glass rod-like structure as seen in this image is the glass electrode, which is dipped in the beaker containing solution, whose pH needs to be ascertained. The image shown in the left lower panel is that of an hot plate, which is usually preset at 40 degrees Celsius. And before we start the procedure, we need to switch on this hot plate as it will require few minutes for this temperature to achieve. The next image is of hybridization chamber. In our department, we have 2 hybridization chambers, and in each of these chambers, we can process 12 slides in each settings. To continue with few more pictures, this is the image of micropipettes having different calibrations. The next image is that of an immunomagnetic sorters. The lower image is a single tube, while the above image is a multi-tube immunomagnetic sorter. We will talk about this equipment in our session of cell sorting. And lastly, this is the image of fluorescent microscope where fish slides are studied for final reporting. Now we move on to preparation of reagents. This is a table showing list of different reagents that are required during different steps of fish analysis. along with their storage conditions. The first reagent is RPMI media. RPMI stands for Roswell Park Memorial Institute. Next is KCl solution, which is used at a molar concentration of 0.075. Carnot's fixative. Wash buffer. We use two types of wash buffer in the form of wash buffer 1 and wash buffer 2. DAPI, which is a nuclear stain. DAPI stands for diamidinophenyl indole. Fish probes and hybridization buffers. Pepsin or protease 2. SSC buffer. Different gradations of alcohol. Chilled water. And few accessory consumables like sealer, cover slip, and immersion oil. However, one should note that this immersion oil is different from the immersion oil we use during routine microscopy. This immersion oil is specific for fish and is commercially available, and it should be fluorescent free. As we can see in this table, most of these reagents are stored at room temperature, especially the reagents that are freshly prepared. Apart from these reagents, few reagents like RPMI, DAPI, SSC buffer, and chilled water are stored at 4 degrees Celsius. Pepsin or protease 2 is stored at -20 degrees Celsius. Most of the fish probes and hybridization buffer are also stored at -20 degrees Celsius. However, some of the fish probes can also be stored at 4 degrees Celsius. This is an image showing all the reagents on a single table. In my subsequent slides, we will be discussing preparation of most of these reagents one by one. Let us move to preparation of reagents. To start with, preparation of RPMI media. As I have said, RPMI stands for Roswell Park Memorial Institute, the place where this media was first discovered. We use two concentrations of RPMI media in the form of 10x and 1x, which is our stock solution and working solution. For preparing 10x RPMI media, we need to add few drops of 1 normal HCl in 50 ml of triple distilled water. In this image, we can see a beaker containing 50 ml of triple distilled water, to which we have added few drops of 1 normal HCl with the help of a sterile disposable syringe to ensure or to avoid any contamination. Next, we dissolve RPMI powder in this acidified triple distilled water, and with gentle shaking, when the whole powder has dissolved, the final color of which will be reddish, we further add few drops of 1 normal NaOH to this solution, again with the help of disposable sterile syringe. The final pH is set at 7.4 to 7.6, which can be ascertained with the help of a pH meter. In the final step, we add 0.5 ml of 10x antibiotics. Different combinations of antibiotics can be used, which I'll discuss in my next slide. Once we have added 0.5 ml of 10x antibiotic with the help of a sterile syringe, and after proper mixing, when we see that the color of the solution gives a pinkish hue, as we can see in this syringe, we can be sure that our 10x RPMI media is ready. Next, we label and store this media in a sterile container for further use. The temperature of storage of this 10x RPMI media is minus 20 degrees Celsius. And as I have said in the beginning, to avoid any contamination, ideally this RPMI media should be prepared in a biosafety cabinet level 2. Next, we move to preparation of working 1x RPMI media. Here, we dissolve 1 part of 10x RPMI media with 9 parts of autoclaved distilled water to ensure 1:10 dilution. To this diluted solution, we add 1% 1x antibiotics. Different combinations of antibiotics can be used. In our department, we use penicillin and streptomycin as the antibiotic. Once this antibiotic is dissolved in this diluted solution, we get our 1x RPMI media. To further ensure contamination-free RPMI media, we need to filter this RPMI media with the help of special filters as shown in this image. This is a single-use filter which has a calibration of 0.22 micrometers. This is the image showing how we use this filter. This one-time use filter is set on the top of the bottle, to which a syringe containing our soap prepared 1x RPMI media is drawn, and with constant pressure over the piston, we pass this media gently over this filter to get the filtered RPMI media collected in this sterile bottle. If we carefully observe this image, we can see that a small part, roughly 1 ml of air is drawn in this syringe. And this has been done intentionally to avoid any contamination and to avoid that at any point of time, the piston of the syringe doesn't come in touch with this RPMI media. This is another image showing the ongoing process of filtration. And one should take care that this air that was intentionally drawn should not be pushed in this filter, because this will stop the filter from further working. The process is repeated again and again until we get the entire filtered 1x RPMI media. And in the last step, we can push this air into the syringe, making it not usable in future course. Once we have filtered this 1x RPMI media, we allocate it in 5 ml quantities in different Falcon tubes, label it, and store this prepared 1x RPMI media at a temperature of 4 degrees Celsius. Next, we move on to preparation of Carnot's fixative, which is a very important reagent in the fish. To prepare Carnot's fixative, 2 major ingredients required are glacial acetic acid and methanol. We take 1 part of glacial acetic acid, to which we add 3 parts of methanol in the ratio of 1:3. We dissolve this mixture to get our final Carnot's fixative. This fixative should be freshly prepared and should be stored at room temperature. Any leftover Carnot's fixative needs to be discarded. Next, we move to preparation of KCl. As already said, we use KCl solution in the concentration of 0.075 molars. To prepare this concentration, we dissolve 280 milligrams of KCl powder in 50 ml of autoclave distilled water, which is pre-warmed at 37 degrees Celsius. The sole purpose of pre-warming this distilled water is it will aid in dissolving KCl powder. Once we have dissolved this entire volume of powder, we have our desired KCl solution. Like Carnot's fixative, this solution also needs to be freshly prepared, and it is also stored at room temperature. Next, we move to preparation of SSC buffer. We use two concentrations of SSC buffer in the form of 2x and 20x. 20x SSC buffer is our stock solution. SSC stands for saline sodium citrate buffer. For preparing 2x SSC buffer, we need to add 66 grams of SSC powder in 150 ml of autoclave distilled water. For ensuring proper dissolving of SSC buffer, we can use this hot plate with the help of magnetic stirrer. Once this SSC powder has fully dissolved, we make the final volume up to 250 ml to get our desired 20x SSC buffer. So prepared 20x SSC buffer is stored at a temperature of 4 degrees Celsius. Next, we move to preparation of working SSC buffer. Here we dissolve 1 part of 20x SSC buffer in 9 parts of triple distilled water to ensure 1:10 dilution. For example, we dissolve 50 ml of 20x SSC buffer in 450 ml of triple distilled water, making the total volume to 500 to get our desired 2x SSC buffer. This SSC buffer should also be stored at 4 degrees Celsius, and the final pH needs to be set at 7 to 7.5. Next, we move to preparation of pepsin. To prepare pepsin, we need 0.1 normal HCl. For preparation of 0.1 normal HCl, we need to add 82 microliters of concentrated HCl in 100 ml of autoclaved distilled water by thorough mixing. The final pH should be set at 2. Next, we take 50 ml of this prepared 1.1 normal HCl in pre-warmed condition, to which we add 25 milligram of protease 2. Once we have dissolved this protease 2, we will have our pepsin reagent ready. Pepsin reagent should also be freshly prepared. Next, we move to preparation of wash buffer. We use 2 types of wash buffer in the form of wash buffer 1 and wash buffer 2. For preparing wash buffer 1, we dissolve 10 ml of prepared 2x SSC buffer in 40 ml of triple distilled water. This makes the total volume to 50 ml with a concentration of 0.4x SSC. We gently warm this solution in water bath, as this will help to dissolve the detergent quickly. Next, we add 150 microliter of detergent in the form of NP40 or IGPAL CA630. And as this detergent is a thick viscous fluid, we generally use mutilated micro tips, as it will ensure free flow of this thick viscous fluid. We mix this detergent by gentle mixing to get our desired wash buffer 1. This wash buffer 1 should be freshly prepared and should be kept in water bath before use. Next, we move to preparation of wash buffer 2. For preparation of wash buffer 2, we dissolve 50 ml of 2x SSC buffer, which is pre-warmed. To this pre-warmed 2x SSC buffer, we add 50 microliters of same detergent using mutilated micro tips, ensuring full dissolvement of this detergent in the solution. We get our wash buffer 2, and we should store this wash buffer 2 solution at room temperature till further use. This should also be freshly prepared. And before I end, I would like to say that out of many variables that can affect the success of a fish analysis, preparation of quality reagents should be considered as one of the most fundamental requirements. I would like to say or re-emphasize the fact that for successful fish, we need to prepare most of this reagent freshly and strictly maintain the pH of these reagents. And lastly, I would like to end by saying that reagent preparation is very crucial part of the entire fish processing, and these reagents act as real agents to solve the clinical puzzle under question. Thank you.